For almost 20 years, scientists have known how to erase the identity of an adult cell and return it to a stem cell-like state. The process helped transform regenerative medicine and earned Shinya Yamanaka a Nobel Prize. But using the same idea inside a living human seemed far too dangerous. Reprogram a cell too much, and it may forget what it is supposed to be. A skin cell could lose its identity. Normal tissue could grow uncontrollably. Tumors could form. In June 2026, that boundary was crossed for the first time. A patient received an experimental gene therapy called ER100. The treatment was designed to make damaged cells in the eye behave more youthfully without completely turning them back into stem cells. It is the first human clinical trial of a cellular rejuvenation therapy based on partial epigenetic reprogramming. This is not a treatment intended to make someone live forever. It is not being injected throughout the body, and researchers do not yet know whether it will restore vision. The trial is primarily testing safety. But if the method works, it could provide the first direct human evidence that some forms of age-related cellular decline are reversible. The treatment targets two diseases that damage the optic nerve: open-angle glaucoma and non-arteritic anterior ischemic optic neuropathy, usually called NAION. The optic nerve is the cable connecting the eye to the brain. It contains the long projections of retinal ganglion cells, which collect visual information and send it toward the brain. When these cells are damaged or die, vision can disappear permanently. Glaucoma often develops slowly. Pressure and other stresses gradually damage retinal ganglion cells, sometimes without obvious symptoms until significant vision has already been lost. Existing treatments can lower eye pressure and reduce further damage, but they generally cannot restore nerve cells that have already stopped functioning. NAION can be more sudden. Blood flow to part of the optic nerve becomes insufficient, damaging tissue and causing rapid vision loss. There is currently no widely accepted treatment that reliably reverses the damage. ER100 is attempting something fundamentally different. Instead of only reducing future injury, it aims to restore function inside cells that have already been damaged. To understand how, we need to separate genetics from epigenetics. The DNA inside most cells contains essentially the same genetic instructions. A nerve cell, liver cell, and muscle cell carry nearly identical DNA. What makes them different is the pattern of instructions they use. Epigenetic systems help control those patterns. Chemical tags, DNA packaging, and regulatory proteins determine which genes remain active and which are silenced. A retinal ganglion cell maintains its identity because it repeatedly follows a specific set of genetic instructions. As cells age, this regulation becomes less precise. Some genes that should remain active become quieter. Other genes may activate at the wrong time. Cells can lose part of their ability to repair damage, manage stress, and maintain normal function. One theory of aging proposes that part of this decline comes from a gradual loss of epigenetic information. The DNA sequence may still be present, but cells become worse at reading and using it correctly. Cellular reprogramming attempts to restore a younger pattern of gene activity. The original method developed by Yamanaka used 4 transcription factors: OCT4, SOX2, KLF4, and MYC. Together, they can push mature adult cells back toward pluripotency, a flexible state similar to that of embryonic stem cells. That complete transformation is useful in a laboratory. Scientists can take an adult cell, create an induced pluripotent stem cell, and then guide it to become another type of tissue. Inside a patient, complete reprogramming would be extremely risky. Cells could lose their identity, produce abnormal tissue, or form tumors. MYC is especially concerning because it is closely connected to cell growth and cancer. ER100 uses only 3 factors: OCT4, SOX2, and KLF4. Together they are known as OSK. The treatment leaves out MYC and aims to expose cells to the factors only temporarily. This is called partial reprogramming. The goal is to move a cell backward far enough to recover younger functions, but not so far that it forgets it is a retinal ganglion cell. The therapy uses an adeno-associated viral vector to deliver genetic instructions into cells in the eye. These instructions allow treated cells to produce the 3 OSK factors. However, the system is designed so that their expression can be controlled using doxycycline, a commonly used antibiotic. In the trial, ER100 is delivered to one eye. The patient then receives doxycycline for a limited period to activate the reprogramming factors. When doxycycline is stopped, their expression is intended to switch off. This control mechanism is crucial. Reprogramming is not something researchers want to leave permanently active. The Phase 1 trial plans to study adults with open-angle glaucoma, or NAION. At least 2 dose levels are expected to be evaluated, and participants will be monitored for safety, tolerability, immune reactions, and changes in visual function. Long-term follow-up is particularly important because gene therapies can create effects that appear months or years after treatment. The trial is not designed to prove that ER100 reverses human aging. It is too small, and it focuses on specific cells inside one organ. The first question is much simpler: can partial reprogramming be performed safely in a human? That remains uncertain. The excitement surrounding the trial comes largely from earlier experiments in animals. Researchers previously reported that activating OSK in retinal ganglion cells could restore more youthful patterns of gene activity and improve function in mouse models of optic nerve injury and glaucoma. Later studies in non-human primates produced results that supported moving toward a clinical trial. According to the company developing ER100, local treatment improved aspects of visual function and nerve structure in primates with an injury designed to resemble NAION. Animal results, however, frequently fail to translate into humans. A mouse optic nerve is not a human optic nerve. Experimental injuries do not perfectly reproduce chronic human diseases. Doses that appear safe in animals can produce unexpected immune responses in people. A biological effect measured in a laboratory may not lead to meaningful improvements in daily vision. The trial could show that the therapy is unsafe. It could be safe but ineffective. It could improve cellular markers without helping patients see better. It may also require years of follow-up before researchers understand the full risks. Cancer is one concern, although removing MYC and using controlled local expression may reduce it. Researchers must also watch for inflammation, immune reactions to the viral vector, unwanted changes in gene expression, damage to the retina, and excessive reprogramming. Even successful treatment in the eye would not mean the same approach can safely rejuvenate the whole body. The eye provides several advantages for an early experiment. Treatment can be delivered locally rather than circulating through every organ. Doctors can directly examine the retina and measure visual changes. One eye may be treated, while the other provides useful comparisons. The amount of therapy can also be kept relatively small. Applying partial reprogramming to the liver, heart, brain, or entire body would be much more complicated. Different tissues contain different cell types. They age through different mechanisms and face different risks. A treatment that helps nerve cells may not be appropriate for muscle or blood cells. Delivering reprogramming factors throughout the body could create uncontrolled effects in millions or billions of cells. This is why headlines describing ER100 as a drug that reverses aging are premature. The trial is testing an experimental treatment for optic nerve diseases. Researchers are not measuring whether participants live longer, become biologically younger throughout the body, or avoid other age-related illnesses. Still, the trial represents an important change in the philosophy of medicine. Most treatments for age-related disease attempt to manage the consequences of damage. Blood pressure drugs reduce strain on blood vessels. Glaucoma medications lower pressure inside the eye. Anti-inflammatory treatments reduce harmful immune activity. Partial reprogramming tries to repair the cellular state that has been lost. If an old or damaged cell still contains the information required to function youthfully, perhaps medicine could teach it how to use that information again. That possibility has attracted enormous investment. Companies and academic laboratories are developing different approaches to cellular rejuvenation. Some use gene therapy. Others are studying messenger RNA, proteins, chemicals, or shorter pulses of reprogramming factors. The central challenge is control. Scientists need to identify the exact point at which a cell recovers useful functions without crossing into a dangerous, immature state. The ideal treatment might need to operate differently in every tissue and possibly in every patient. Researchers must also determine what rejuvenation actually means. A younger epigenetic pattern does not automatically equal a healthier cell. Biological age markers can change without proving that tissue functions better. A treatment should ultimately preserve vision, restore strength, reduce disease, or improve survival, not merely make a molecular clock display a smaller number. For patients with glaucoma or NAION, the immediate question is whether damaged retinal ganglion cells can recover enough function to improve vision. For aging researchers, the trial asks something broader. Is cellular aging a one-way journey, or can living human cells be guided safely toward a more functional state? We do not yet have the answer. Only the first participant has crossed the starting line, and Phase 1 trials are designed to discover risks rather than confirm dramatic benefits. The outcome may be disappointing. Many experimental therapies fail. But for the first time, partial epigenetic reprogramming has moved beyond cells, mice, and monkeys into a human clinical trial. Scientists are no longer only asking whether old cells can be made young again. They are beginning to test it.